Hello, I'm Jesus de la Fuente. I'm a first year PhD student under the supervision of Dr. Miguel Hernandez and Dr. Ideocho. And today we're going to be presenting the identification of transcriptional network disruptions in drug-resistant prostate cancer with TREAD. So TREAD stands for transcriptional rewiring. It's a computational tool which allows to identify phenotype-dependent gene regulatory networks. It was developed in the context of PROMOTE, which is a Mayo Clinic clinical trial. It's a metastatic castration-resistant prostate cancer study from which we have a bulk RNA dataset containing 21K genes and 46 patients. These patients were sequenced before they were given abiraterone, and responsiveness was measured to generate the associated phenotype. TER was developed to uncover gene regulatory network disruptions among a binary phenotype, which in our case was responders and non-responders. From a given matrix containing the gene expression counts for n samples, GRNs are discovered in two phases. During phase 1, target genes are iteratively partitioned into K modules, where their expression patterns are modeled by a sparse set of related transcription factors, what we call regulators. After this, a variational bias spiked regression model is used to explain the target gene expression as a linear combination of driver genes in order to infer gene regulatory networks. The inferred gene regulatory networks are then evaluated against the rewiring test, where the significance level of network disruption is measured by the Frobenius norm of the differences of the covariance matrices among responders and non-responders. For so, a permutation test is applied to every inferred gene regulatory network, and if the true phenotype associated gene regulatory network is significantly disrupted among a fixed amount of permutation, the GRN will be labeled as rewired. Note that the yarn inference and the rewiring scoring process will be repeated b times using different subsets of samples in order to account for robustness. So from the rewired inferred GRNs across runs, a hierarchical clustering technique groups together similar rewired gene regulatory networks, yielding robust rewired GRNs. On the other hand, drivers within the rewired GRNs are sorted according to their specificity among rewired or not rewired GRNs. We consider a driver to be rewired if it appeared a significantly larger number of times in a rewired module than in a non-rewired one. So for this purpose, we increased the number of runs up to 50 and computed a p-value based on a hypergeometric test from the associated table. So when the GRN inference process was applied on synthetic data, we showed that in fact TRER builds true modules and identifies rewired GRNs on synthetic data. For so, we generated 10 synthetic GRNs and applied the GRN's inference process 5 times. Here we see how synthetic GRNs are correctly uncovered within each run and group them together according to a clustering technique to recover the original synthetic GRNs. On the rewiring side, 3 synthetic modules were originally enforced to be rewired, and again TRED correctly uncovers them and groups them together across runs, yielding robust rewired GRNs. So when TRED was applied to the aforementioned PROMOTE clinical study, a robust rewired GRN was uncovered, which contains a clear disruption from one phenotype to the other one. Also, when applying a clustering technique to the inferior GRNs from the PROMOTE clinical study, 93 sets of highly overlapping GRNs were discovered. 11 of them contain one or more rewired GRNs. So we applied the Community Amaretto software to functionally annotate them using gene set enrichment analysis, and we focused on the Community 22 as it was fully composed of rewired regulatory networks. So we generated a list of transcription factors from the rewiring at the gene level within the Community 22, and we decided to explore the effect of these transcription factors in the cytokine-chymokine pathways. And after knockdown of the top transcription factor, the expression level of these genes were found to be significantly changed by the quantitative real-time PCR of two pairs of parental and Navi-resistant prostate cell lines. So this has been the identification of transcriptional network disruption in drug-resistant prostate cancer with TERN. I hope you have enjoyed the short talk, and I will be really glad to answer any question you have. Thank you very much. I hope you have a good day.